What we've talked about so far, we've already talked about our neurons. We talked about the generic structure of the neurons. We talked about the excitability of neurons. So by now you guys are experts on the mechanism as to how we get these neurons to fire, with the end goal that neurons can communicate with one another. We've talked about the synaptic terminals, the release of neurotransmitters, excitatory neurotransmitters, and inhibitory neurotransmitters. And what we're going to be doing is tying that in. So when we looked at the central nervous system, now we know all of the functions of the nervous system. We know, looking at the different regions of the brain, where we saw regional specialization in terms of function. But what we're going to be focusing on in the peripheral nervous system are all the nerves, all the axons that project out of the central nervous system. So with that, let's start to go into our first slide, which is... Have I talked to you about the Body Worlds exhibit?
No.
Oh God, you guys gotta go to it. Okay, so the Body Worlds exhibit it's a great opportunity to go. The last time I went was about 13 years ago, and that was I went on a first date at the Body Worlds exhibit, and I totally geeked out. And what was great about it too was because it was like the last week. It was one of those things where it was open like 24 hours. Because there's so many people, and it was like an excuse to be out like 3 in the morning. Let's go look at some dead bodies at 3 a.m. But nonetheless, it's out right now. So it's a guy named Dr. Gunther von Hagens who created this process called plastination. And upon death, what happens is that people, before death, people will will their bodies to. You guys know how you can will your bodies, like donate your bodies to medical science. You can donate it to any organization, and one of his organization is specifically this. It's the process in which he takes willed bodies and he plastinates them. Basically, he dehydrates your body and he injects this liquid polymer in there, and then he goes through the process. And people, anatomists in his lab, take the time to dissect the cadavers into specimens that show things like this. They want to show specifically all the nerves. The central nervous system shows the brain, the spinal cord, and all the nerves, and very thorough dissection too. These are like experts in dissections. And then they plastinate it. They basically bake it so that it turns into this hard plastic. And they get posted up as his exhibits. And so what Dr. Gunther von Hagens, when he first created this process, was back in Japan, I think back in the 80s or 90s. And these were willed bodies that were hung like this, and he had them on like hangers, and they were hanging by their necks. And it was an exhibit for people to come in and take a look at what the human body looked like. And they had a line that wrapped outside the door because they were interested. People walked in, looked at like a first couple exhibits, and they walked out. They were like, no, this is disgusting. And so he realized at that point, okay, this is not really any interest in the human body. Because his interest is anatomy. He wants to show people what the human body actually looks like. This is really, and the reason why universities, colleges want you to do a real dissection is to really take a look and see what it looks like. And so what he wanted to do next was to kind of venture more into art and to put the bodies into something that looks a little bit more lifelike and in easier words, looks a little bit more pleasing to the eye. So he puts people in these configurations. There's a guy playing chess where he's literally sitting down, he puts the eyes in, you can see the brain, and he opens up the skull cavity so you can see the brain as is. He shows people playing basketball, and he has a bodybuilder that has all the muscles, all the skin removed, and he's holding up the skin, and you can see the amount of muscle that this specimen specifically has. And so he bridges this whole science and art. And what it's done is it's attracted people to come want to look at these things. Because, I mean, I've been there a couple times now. You see kids that are going in there just completely marveled. They're like, oh, this is what it looks like. It's easier to look at than it is to look at specimens on a table. So with that, it's out at the California Science Center, I believe. And it's out till February. So it hasn't been around here, and he rotates his exhibits all around. He won't see it probably for another decade. So I was glad when it was out because I was out there, and to see the new specimens that are out there, you learn quite a bit. You get to really see what things look like. If you do go, there's the first floor, which is kind of more mediocre. You can see like things in a table. They'll show you what the stomach looks like. They'll show you what like a hip replacement looks like. They show you just basically some of the muscles that are involved. And then they give you the ticket to go upstairs, and that's where you can start to see more of the heavier stuff. You can see entire specimens. I know I remember seeing a horse that he had dissected, uses other specimens involved. Great opportunity for learning. I assure you, if you take an anatomy or you just have keen interest in looking at the human body form, please go by all means, take a look at it. So now that I've digressed from all that, this is one of his exhibits, just showing specifically. You can kind of see the outline for the other specimens in the back. But this dissection, he specifically went through and dissected each of, obviously the brain and the spinal cord. That was easier, but then he goes through and takes out all the nerves. When you guys did your dissections, you saw all the highways, basically your artery, your veins, and your nerves. So the question that we're going to go into later, what exactly are the nerves? So when we look at it from a functional standpoint, we'll be able to look at the first division, which is the somatic and autonomic nervous system. So looking at that, this is our breakdown where we can see what we've talked about so far. We've talked about the entire left side of this chart, the central nervous system. We've talked about the functions, we talked about the brain, we talked a little bit about the spinal cord, we went to the forebrain, midbrain, hindbrain, cerebrum, all the way down to the medulla. Now what we're going to be talking about is the peripheral nervous system, everything outside of the central nervous system, of which we can break down, again, somatic nervous system and autonomic nervous system. Now the autonomic nervous system, when we take a look at it, you can see we're going to be able to break that down to sympathetic and parasympathetic. That we talk about on Tuesday. Today, we talk specifically about the somatic nervous system, afferent nerves and efferent nerves, but really the focus is on efferent nerves. What exactly is the somatic nervous system? We're talking about the connection from the central nervous system out to the muscles. Okay, somatic meaning body or muscle control. Autonomic, this is a breakdown of things where we're looking at things that are autonomically regulated. This would be like your heartbeat and such. Again, I'll talk about that in the next couple slides. So a couple things to take a look at before we get into the heart of what we're going to be talking about. A couple terms I want you guys to be familiar with. When we think about neurons, neurons aren't just scattered around the human body just randomly. We have them already organized as such. And here you can see these different highways of nerves. When we look at these neurons, again, neurons, the nerve tissue is basically a whole cluster of neurons that work together. So we have to think about what these clusters look like. And the first thing we're going to be looking at is the term ganglia. And ganglia is a reference to neuron cell bodies that are together. Again, cell bodies that contain the DNA and where they're located. And again, I'll pinpoint where that takes place on the spine. Now nerves, those nerves that you saw when you did your dissections, again, you saw arteries, you saw veins, and you saw nerves. What color were the nerves? Do you remember?
Purple.
Oh, red maybe because of a little blood, and purple, those were the veins. Right? There were white strings, right? It kind of looked like, and there's always that person that says, it looks like some food item. It always looked like string cheese.
Tendon.
The nerves, they were white, and why were they white?
Myelinated.
Myelination. So nerves, by nature, when you look at them, are covered in myelinated tissue. That is the myelinated axons. So nerves are bundles of axons. Anytime you looked at a nerve that projected out of any of these nerves that you see right here, these spinal nerves, you see those. Those are projections of axons that travel to their muscle destination. So think of it as a highway going to its target muscle in order to get that muscle to contract. The neuroglia that are involved, satellite cells. So satellite cells were the ones that wrap the cell bodies in the ganglia. We'll be able to see that in a bit. And Schwann cells were the ones that covered the peripheral axons in myelination. So looking at this here is going to be the first, and we're going to revisit this over and over again. We're going to be looking at the pathways, receptors that receive information. Remember, this is the sensory portion. Receives information, sends that back over to the central nervous system, specifically the spine, travels to a specific area in the brain. Sensory information is going to travel to the sensory motor cortex. Okay? And again, we'll revisit that in a little bit. And then sends efferent information. Remember, all the interneurons here are processing that information, processing that sensory information, which we call perception. and then sends that motor command back out. This is the efferent pathway. So information goes into the central nervous system, comes back out the peripheral. And we'll talk about those. Everything that leads out of the central nervous system to the target muscle. Again, satellite cells, when we look at our ganglions, here you can see the cell bodies, and you're going to see all the littered satellite cells that surround an area for the purposes of regulating the extracellular fluid. It wants to make sure it has the right amount of oxygen, get rid of that excess CO2, make sure you're providing glucose, nutrients, and everything else to ensure proper functioning of that neuron. The Schwann cells, on the other hand, are the ones that wrap the axons. While these work around the cell bodies, Schwann cells work around the axons themselves. So it wraps around and creates that myelination. So if we have myelination on an axon, that's going to be working off saltatory conduction. Unmyelinated axons are going to be continuous. And there's going to be relevance behind that. So looking at the efferent pathway, again, pathway from the central nervous system out. You can see we have somatic and autonomic nervous system, in which the somatic nervous system is responsible for voluntary, right, control of skeletal muscle. So these are things that you have voluntary control of. Autonomic nervous system are things that you have involuntary control of. Right? So things, and again, going back to that initial neurophysiology lecture where we did that exercise looking at that guy that almost got hit by the car. We talked about things that he was able to control, to jump out of the way of that moving vehicle. But what other things did he not have control over? His breathing, sweating, heart. Okay? So those things he doesn't exhibit control. Those are autonomically regulated. So involuntary control over cardiac and smooth muscle. So this is typically related to the visceral organs, while somatic is control of skeletal muscle. Now the key portion of what you're looking at here for somatic is that it says part of the autonomic nervous system. So the question is, why do we have voluntary control over some autonomic systems?
Yes.
Breathing. So if I ask you to start, normally you're not thinking about breathing, but if I all of a sudden say, I want you guys to breathe a little bit faster, sure, you can take control over it. Okay. You do have some control exhibited over your heart rate. So if I tell you, control your heart rate right now, you may not be able to say, okay, heart, slow down, but you can put yourself in a position to relax and control and bring your heart rate back down. You can do something also with salivation. You can trigger salivation. You can learn salivation. You can actually start to learn to salivate. You've probably seen people in elementary school that can do the little spit things that they can do. They can literally shoot spit out of the glands. And then also something that we don't really talk too much about, but sexual arousal too. So that is something that you do have voluntary control over. The part that's actually autonomic is orgasm itself.
But I heard that they say some people, they say you cannot stop the breath to that.
Well, I'm sorry,
Eventually,
what are we talking about?
The breathing. You say
breathing,
you can control it, but when you try to stop it, like not to breathe until you're dead, you can't.
No, you can't.
Your body gonna kick in and then you're gonna breathe, right?
Yeah.
Okay.
So that part you do have voluntary control over, but there's something else that will kick in that will override that. And that we talk a little bit more about in respiratory.
Okay.
So with that, let's take a look at the cranial nerves. And here you see all 12 of them. Okay? I already said before, you're not going to have to memorize them. It's still true, you don't have to memorize them. We will talk about a couple of them. They're still considered cranial nerves because they are nerves that project out of the brainstem. And then the spinal nerves being the nerves that project out of the spinal column. But a couple of them that we're going to be talking about, we'll be talking about, let's see, vagus is one of them, cranial nerve 10. We'll talk a little bit about the glossopharyngeal when we talk about our sensory lecture next Thursday, at least when I upload that on YouTube. And we'll talk a little bit more about vestibulocochlear, again, when we talk about hearing. But really, the main idea is we know these are projections out of the central nervous system. So 12 pairs total, again, not required to remember the names. You already did that in anatomy. The next thing is to take a look at the spinal nerves. We'll be looking at specific slices along just the generic spinal cord. And so what we're looking at specifically is the dorsal. This is the dorsal area, the back, posterior. And this is the ventral portion. So this is more on the interior, more the anterior side. So what you're looking at here is the spinal column. And if this is the dorsal side, and this is the ventral side, we're going to be looking at the gray matter that's on the inside. Again, gray matter because it contains the cell bodies. White matter because it contains the myelinated axons. So we're going to expect to see axons travel through the white columns that you see. Again, more details on that. But we're looking at the projections that come off the central nervous system. So we know that the projections, and we'll learn that the projections come out. You'll be able to see there's a dorsal root and a ventral root. And those come together right in between. And then the spinal column itself travels to the vertebral foramen. So right in between each right here, as you can see on our spine, you'll see that's where our spinal nerve comes out. Okay? So the dorsal root and the ventral root come together, and then they project back out. And this is where you're going to start to see the divisions. This is the efferent pathway that we're talking about. If you're wondering where's the afferent pathway, remember what we talked about at the end, the last thing that we talked about prior to the exam is the pathway. Sensory information travels into the dorsal root, into the dorsal gray horn, out into the ventral gray horn, and back out through the ventral root. So pathway travels this way, in, through, and then back out. So really what this comes down to is the implications behind this is if you were to look at a spinal fracture of any kind. The question is, where does the damage occur? Does the damage occur specifically on the spinal nerve, which would sever all afferent information and efferent information? Do you sever the ventral root, which would only remove the efferent pathway out? Or do you damage the sensory portion, more the dorsal root, which would affect any sensory information going out? Okay, so typically that's something that they look at in terms of the fracture. But if you were to cut at the spinal nerve, you remove all afferent and efferent information going in. Now looking at this, so we have our spinal nerve, just like I pointed out earlier, our spinal nerve, and then we see the projections out, because this is where we want to split those nerves out from that particular spinal cord section. Because now we want to look at the direction of where we're going to travel. Anything that travels this way is probably going to be on the dorsal side, somewhere on the back. Anything out on the side, and we're going to be able to take a look at that in just a little bit, the ventral side is anything that's in front of the spine. So everything antero, everything in the anterior and lateral regions it's going to target. And then we'll talk about this. This is going to be the branch for our autonomic pathways. So when we look at those spinal nerves, of which we have 31 total, these are the branches that we're looking at. Just like you saw over here, we're not going to twist it back up, and you're going to see the projections out here. Same projections that you're going to see out on this right here. Now, you'll see that the term that's common with all of these, once we split from the spinal nerve, here we have the dorsal root, we have the ventral root. They come together, the spinal nerve. Right here is the intervertebral foramen. Now, where we split back out are called rami. That's the term that we're looking at, rami. Rami means branch. So we're looking specifically at the pathways that branch away from that spinal nerve. And looking at this, we can see where those branches go. So the first branch that we take a look at is the dorsal rami, or dorsal ramus. This is the area that's receiving and sending motor information to the skin of and muscles of the back. So everything behind the spinal cord. The ventral ramus, if you take a look, here's our dorsal ramus. This basically targets all the muscles in the back. The ventral ramus, you can see, is actually the biggest one. That's the biggest branch that you see on here. And what that targets, the ventrolateral. So everything in front and to the side, body surface, including not only the trunk that you're looking at, but also the muscles of the upper and lower limbs. So the extremities. And then the meningeal branch, we don't even really talk about that much. There's one branch that comes out. It literally comes out and then runs back in and targets the meninges. That we don't really talk about that much. But the last one that you see, so if this is ventral, this one that comes out is the rami communicantes. And this is what it will travel out and then back in. We'll talk about this next Tuesday. What we're talking about are, again, things that are related to somatic nervous system, which are going to be our dorsal and our ventral ramus. So when we look at these, now that we've looked at a generic slice of the spinal cord, now we're going to start to arrange them all the way from cervical to lumbar to thoracic, cervical to thoracic to lumbar to sacral. And when we stack these up, we look at where these spinal nerves directly innervate, whether they directly innervate or indirectly innervate. This slide answers the question of a spinal nerve that travels out from a particular spinal vertebra. Where does it target? Does it directly target a muscle, or does it go to a specific highway? And what do I mean by a specific highway? I'm going to go back to our first slide. Here you can see, if you're ever wondering, where are the nerves traveling from this area? Why does it look like this area, it's about T2 to about T12, where are the nerves on that side? Where do they go? These typically will travel directly to the muscle. From the spine out to the muscle itself, it's a direct nerve that travels. Everything else, everything from the lumbar regions, what you notice about these is they start to kind of converge back together. You guys see that? They converge back together and then they split back out. So this is a way to consolidate those nerves to make their way into what's called a plexus. So going back to that slide, we talk about direct innervation and indirect innervation. Direct innervation means T2 to T12 target directly to that skin and to the muscle. Now, indirect innervation means that the nerves that travel out, and the major ones that you're seeing, we're going to see these plexuses. The nerves that travel out the cervical region, as well as the sacral regions, travel directly to what's called the plexus. And that plexus is a network of rami that join together. Think of all those highways that travel out of those spinal nerves that converge, and they merge all. Think of it like a bunch of freeways merging all into one freeway. And then, once they reach that plexus, they're going to merge. They're going to diverge from that area and travel out their separate ways. The four main areas that you're going to ever see the major plexuses, or plexi, I believe: cervical, brachial, lumbar, and sacral. And let's take a look at that. What that looks like. Here you can see from this diagram, you can see here's T2 through T12, and they go directly to the thorax itself. All the other ones converge into the plexuses that you can see. So let's take a look at what they look like. You can see all four plexuses from this slide. Top left, here you see all the cranial nerves that come out. You have cranial nerves and you have some of the cervical nerves. And C1 through C5 come together. C1 through C5 come together into this region, this plexus called the cervical plexus. And those target specifically the skin and the muscles of the head, the neck, as well as the diaphragm. So that's our first area. Again, a whole cluster of nerves. If you were to take a look at the next one, the brachial plexus, this one's the one that is the most branched. And this starts from C6 to about T2. So C6 to T2. There's a whole region of nerves that all come together to make their way, and then you get these three plexuses, major plexuses, and again, then they branch right back out. So that way it all comes together and comes right back out. The next one, you can see the lumbar plexus, and this lumbar plexus is from L1 to L4. You'll see that they project out as a spinal nerve, and then they come together. They merge together on the freeway, and then they come right back out and split right back out. And then the last one is the sacral plexus, L5 to S4. Those come together, converge together, and then they branch right back out. So common areas that you would see with sacral regions, this is the area that typically governs the lower back and the lower extremities. They come together. And one area in particular that's common, a common injury, is sciatica. Does anybody know anybody with sciatica? Yeah, so this is chronic back pain. And that is really related to one of the nerves that actually comes over and travels right by the piriformis. And that muscle, when inflamed, pinches along one of those nerves that come off the plexus. That causes that shooting pain that they feel. So that's something that you typically see on here. The other things that there's other smaller plexuses. If you ever heard of the solar plexus, or if you were ever to get into, for instance, I wouldn't advise this, but if you're ever to get into a fight with somebody, you target the plexuses. Those are the nerves. Those are areas that if you were to cause any damage to, causes quite a bit of nerve shock that takes place. Okay, so those areas on the side of the neck, brachial, lumbar, and then sacral. So looking at that, now that we have the anatomical highways that travel outside of the cranial nerves as well as the spinal nerves, let's hone in back and take a look at just the functions of them. Looking at the somatic nervous system, again, the voluntary aspect, the stuff that we already learned, is sensory information traveling in. Again, something that we talk about next Thursday. The central nervous system, which we already cover, which is the interneurons, the integrative center, that's going to integrate all that sensory information, process it, and make some sort of decision. It can also store it as memory, it can make any sort of decision making, and then send that path back out, the motor pathway, to the muscle itself. So in the somatic nerve pathways, this is the efferent pathway, of which we're going to be really focusing on today. Integration and then motor pathway. We'll circle back on sensory next Thursday, looking at how information travels to the central nervous system. Now, looking at the pathway, when we look at the pathway from the central nervous system out to the target muscle, the key characteristic of this is that the pathway from the gray matter of the spine, which is going to house the cell body, is going to project its axon all the way down directly to the muscle. So this involves a single motor neuron. So if you think about the connection for your muscle, let's say whether you're looking at your quadriceps, everything from that connection, the cell body, the axon travels that entire length. And at the very end of that axon, the synaptic terminals at the end are going to secrete acetylcholine. Acetylcholine is, and we'll talk about that in the next slide, is that neurotransmitter. And that the effector at this point is skeletal muscle. So if you ever think about paralysis, the inability for motor control, to exhibit motor control on a target extremity, on any muscle, is specifically that. Did any damage happen along that axon, along that pathway, along those nerves? So again, one single motor neuron system. Later on, when we talk about the autonomic nervous system, you're going to see that there's actually two neurons involved. But again, we'll save that for Tuesday. So single motor neuron. And where is the cell body located? Cell body is always located in the ventral gray horn. For autonomic, what we're going to learn, it's in the lateral gray horn, off to that little bump to the side. But right now, where we're looking at these motor neurons, that motor neuron has a specific location right here. This is the location for one muscle. This is another location for another muscle. This is a location for another muscle. All the cell bodies reside within that ventral gray horn. Now, this slide talks about this junction right here, from the synaptic terminal to the muscle layer itself. This is what we call the neuromuscular junction. And in the neuromuscular junction, this is, by definition, the synaptic cleft. This is everything between the axon terminal and then the sarcolemma. The sarcolemma is the outside of the muscle fiber that has the acetylcholine receptors that receive acetylcholine. So if acetylcholine is secreted by the axon terminals, secreted out into the sarcolemma that's going to contain the receptors for acetylcholine. So this is where this is taking place. Secretion by exocytosis. The acetylcholine will attach to the receptor. And then that receptor allows depolarization to take place. So what we're seeing that's taking place in the muscle is sodium entering in. Okay? The key thing about acetylcholine, let me backtrack. When we depolarize the cell, this will result in reaching threshold of the muscle, and then the elicited effect is the contraction of the muscle. So acetylcholine is only excitatory. The only way we can get a muscle to contract is the release of acetylcholine. We release it, it'll bind to that receptor and cause depolarization, resulting in contraction. So the question to think about is, how do we cause inhibition? I'm not going to answer that just yet. All right, so going back, here's our motor neuron that's located in the ventral gray horn. It's going to send an action potential down and release acetylcholine. What if we don't want that muscle to fire? Okay, what do we do to make this muscle less likely to fire? There is no inhibitory neurotransmitter in between the motor neuron and the muscle fiber. So the only way I can get this muscle to less likely to fire is to go back and take a look at interneurons. And I've got a slide talking about that later on. And what that interneuron is going to do is going to target that motor neuron. And remember what I talked about earlier. This is, by definition, the cell body. Here's my axon. And in order to make this motor neuron less likely to fire, we have an inhibitory neurotransmitter. And what I'm drawing right here is a negative with a circle around it, knowing that this is what we do to cause inhibition. So looking at this, if this is our interneuron, which is located in the spinal cord, hence why it's considered an interneuron within the central nervous system, if that secretes any inhibitory neurotransmitters, right, just like that test question, an inhibitory neurotransmitter on there most likely will cause this to hyperpolarize. And what does hyperpolarization do? It does what? Does it make it more likely to fire or less likely to fire?
Yes.
Less likely to fire. So if I get that motor neuron less likely to fire, that means that muscle is less likely to contract, meaning we decrease the likelihood that we secrete acetylcholine. So what governs whether a muscle is going to fire or not fire? And really what it comes down to is how many action potentials will get fired upon. Remember what happens when we get this motor neuron to twitch just one. If I send an action potential, this causes a little twitch. But if I really want to elicit a long contraction, right? If I have a suprathreshold stimulus, that's going to elicit multiple action potentials being fired. If I want to get that bicep curl, remember if I used that example earlier, if I want to get that bicep curl to take place, I don't want just one muscle twitch with one action potential. I want successive firing of that motor neuron. And that will generate the muscle tension that's needed to continually contract the muscle. Now, if I want to inhibit it, I would have to have an inhibitory interneuron that makes this motor neuron more hyperpolarized and less likely to fire action potentials. And that decreases the muscle contraction. So now that you know that, what relevance are we ever going to be seeing times when we want to turn off contraction? So think about where would we see that? Why would we want to stop a muscle contraction? Okay. We're also going to be able to see reflexes. If a muscle is contracting and a reflex is elicited to turn off that muscle from firing, that's a reflex, and that is a form of negative feedback. And we'll be able to see that at the end of today, reflexes in which a muscle contraction results in turning off the muscle contraction. And I'll have some relevant examples for you when we get to that point. So keep in mind that what we're doing for a muscle is the sum of excitatory and the sum of inhibitory postsynaptic potentials taking place on that motor neuron. It's the summation of all of those. If I want more firing, then get some excitatory interneurons. If I want to inhibit, then I use inhibitory interneurons. And all that summates on that motor neuron that will decide whether that muscle will fire. Does that make sense for people? A little bit? Of course. So the motor neuron is going to ultimately decide if the muscle is going to contract. And the likelihood of that muscle firing is dependent on all that feedback that's being sent to that motor neuron. So interneurons, a combination of excitatory and inhibitory interneurons are going to come in and send postsynaptic potentials to that motor neurons to make it more likely to fire or less likely to fire, depending on whether we're trying to excite that motor neuron or not excite that neuron.
So
Inhibit.
So the motor, the interneuron pretty much tells it has both inhibitors and the ones that excite, or it's just one or the other?
There's actually excitatory interneurons and inhibitory interneurons.
So then if, for instance, it's excitatory, then it's going to send a message to the motor neuron to go ahead and excite it and send the message. But if there's more of the inhibitors, it's going to send, okay,
Got it. So it's the summation. So what you're getting at is summation, what's going on. To give you an example, is a reflex that typically happens. You can, and interneurons aren't just in the spinal cord. We have interneurons up in the brain too. So you're going to be able to see later on, this will hopefully tie in later on, we're going to get some axons that are going to travel in, down from the brain, travel down our white columns. Remember, these are axons that travel through, and they're going to come back to this motor neuron and say, no, don't fire at all. And that's going to inhibit that firing that's taking place. All it comes down to is, if you're ever to just look at muscles themselves, it's just a bunch of either you contract the muscle or you don't. And whenever we perform any coordinated movement of walking, it's a bunch of turning on a muscle, turning off a muscle, turning on a muscle, turning off a muscle. Some muscles are turning on and off, some are contracting a little bit, some are contracting more. It's this coordinated pattern. And what part of the brain is governing all of that? Which we'll learn is the motor cortex as well as the cerebellum. Again, we've got slides that talk about that. Really the heart of all of this, what I wanted to get at, how do we inhibit a muscle from contracting? Was just that inhibitory interneuron. If you got that, you're good for now. What we've been describing are the lower motor neurons. Even though I call it the motor neuron, it's technically the lower motor neuron, because the lower motor neuron targets the muscle directly. These are lower motor neurons. And here again, you can see are located on the ventral great horn. They target directly to the muscle with that single motor neuron system. Whether they're located in the cranial nerves or the spinal nerves, any way you look at it, they target directly to the muscle. That's our lower motor neurons. The next thing we're going to be able to see is that we also have feedback. All that stuff that I've been talking about, all the interneurons that are that play that role on that lower motor neuron, we've got different types. We've got local circuit neurons, also known as interneurons. We have upper motor neurons, we have basal nuclei, and cerebellar neurons. These are all the central nervous system interneurons that come to play on that motor neuron. Again, that motor neuron projecting out the ventral root, that travel out the spinal nerve, that travel down either through direct innervation or through the plexuses directly to their target muscle. Interneurons. Basically what I've been talking about earlier, about the whole process of interneurons, is that they receive, again, looking at interneurons located in the central nervous system, their whole role of interneurons is to receive sensory information. Okay? They receive sensory information, they process it, and then they can determine what motor effect to do on here. So the things that you're going to be able to see, that's where it's receiving. Here you can see our interneurons located in black. This is just one example of interneurons. And here you can see excitation of that motor neuron that targets the hamstring. Here you can see inhibition with an inhibitory interneuron of the quadriceps. So what the interneuron does is it receives information from the muscle itself. This is a form of a proprioceptor. Again, something I'll talk about next Thursday. A proprioceptor sends sensory information about the status of that muscle. It's going to send information about the joint, send information about the tension of the muscle, sends that back through the dorsal root ganglion. Dorsal root ganglion, remember ganglion being a cluster of cell bodies, of a unipolar sensory neurons. It's going to send that information back into the dorsal gray horn and target the interneurons that are located. The interneurons are the ones that are going to coordinate which muscles are going to contract and which ones are going to relax.
So they work together, basically.
They all work together to exhibit that control. So the interneurons here are what's taking place in the spinal column. You're going to see that there's also interneurons located in the basal nuclei, the cerebellum, as well as the primary motor cortex. And they're all going to send projections out onto that motor neuron to govern whether it's going to fire or not. So here we mention excitation. If you have an excitatory interneuron, it's going to depolarize the motor neuron, make it more likely to fire. If you have an inhibitory, just like we mentioned earlier, that's going to hyperpolarize and make it less likely to fire. And all of these are going to sum up and tell us, are you going to fire or are you not going to fire? And if you're going to fire, how much you're going to fire. That's all it comes down to, is to be able to determine the amount of muscle tone and the amount of muscle contraction that will take place. This one talks about their local circuit neurons. These things that I've been talking about right here, these are what are called, so all the things that we've been talking about, interneurons, these specifically are our local circuit neurons. Why are they called local circuit? Because they happen within that spinal cord segment. Right? It's literally a local circuit. This is a reflex. Information from the muscle gets sent back in. Interneurons are the ones that decide, and locally either excite or inhibit a motor neuron, hence local circuit. The thing is about also local circuit neurons is it not only works on specifically this slice, but it can go in and target other motor neurons on other slices. So if this is one slice that you see, think of another slice that's just like another millimeter away. And there's another motor neuron that comes in that may target the same muscle. So what local circuit neurons do is they can recruit all the neighboring motor neurons to say, if I want this motor neuron to fire, I want the other motor neurons to fire as well. And what that's going to do, it's going to target that same muscle and cause an even increased contraction. Not only do I want one motor neuron that targets that muscle, I want all the other motor neurons that target this muscle as well in adjacent spinal cord slices. What's the point in all that? Now you get muscle groups that all work together. This coordinates the rhythmic action. The next thing you can see on the next slide are upper motor neurons. And as you can gather from the name, this starts in the upper area. So where does this start specifically? Let's go back to this slide right here. Here you can see upper motor neurons. And where do the upper motor neurons originate? They originate right in the primary motor area of the cerebral cortex. And if we remember, you have to really dig back in your memory and think about what we saw when we looked at the primary motor cortex. You remember there was a sensory motor homunculus where you saw the face over here, you saw the leg, and there's my foot, and then you saw some of the hands over here. And on this particular slice, remember the primary motor cortex was the more posterior region of the frontal cortex, or the frontal lobe. Okay. So right the front part, right in front of the central sulcus, was the primary motor cortex. And where on that brain, or where on that motor cortex, corresponds to the cell bodies. Again, the outside area represents the cell bodies. Gray area is the cell bodies. So these represent the cell bodies that innervate that spinal cord segment that travels to that motor neuron. So if this motor neuron targets the quadriceps, that area that represents the quadriceps up on that primary motor cortex is the upper motor neuron. And that upper motor neuron projects all the way down. Down, 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 most of it, about 90%, will cross through the medulla oblongata and travel to the contralateral side. Others, the other 10%, will travel down through these anterior columns, and again, I won't ever have you guys memorize which column it goes through. It's just going to travel through that through one of the white columns and cross over through the gray commissure. And so those interneurons will come in and innervate that specific motor neuron. So just to clarify, lower motor neuron is directly to the muscle. Upper motor neuron is from the brain, from the primary motor cortex, to the lower motor neuron. And typically, whenever I want something, if I'm consciously trying to think of a muscle action, if I'm consciously thinking about a bicep curl, I'm thinking about the area that's going to innervate, the area that corresponds to the bicep. That's a upper motor neurons are going to travel all the way down to that spinal cord segment that travels to the motor neuron that innervates directly to that bicep, causing a contraction. Does that make sense?
So does the lower travel to the upper?
No, lower travels directly to the muscle.
Okay.
Upper travels from the brain down to the lower motor neuron, but never backwards. This is efferent information out. So the purpose of this, and this is something that I know I'll talk about a little bit later on. This is typically when we look at our upper motor neurons, this is conscious control over. Lower motor neurons, when we look at interneurons and stuff like that, that stuff, projections that are, I want to still say, are still voluntary control, but don't involve the cerebral cortex. These are the projections that we're going to be able to see that come in from the basal nuclei as well as the cerebellum themselves. Now, looking at the upper motor neurons, you're going to be able to see direct motor pathways, which is, for the most part, direct motor pathways travel from the motor cortex down to the lower motor neurons themselves. So direct is typically excitatory. The other thing we're going to be able to see are indirect motor pathways, extrapyramidal pathways, and these are typically inhibitory. Now, I'm not really going to go into too much descriptions of these, simply because there's so many pathways that travel. If you were to take a look at the projections for the upper motor neurons, they travel down the white columns that are represented here in red. And we have different tracks that you can see on here. You can see corticospinal, and all the ones that are red are the efferent. There's ones traveling down the spine, while the sensory ones, the ones in blue, are axons that project up to the brain. If you were to take a slice of the spinal cord, you can see that there's specific highways. Okay? So the interneurons we talked about, upper motor neurons, there's local circuit, there's the basal nuclei, and the cerebellar. All of those, every interneuron is in the spinal cord as well as the brain. So those upper motor neurons that travel down will travel down these tracks. And again, if we remember, I mentioned I'm never going to ask you guys which travel where, but you should be able to determine. If I were to tell you a spinocerebellar tracks, spine to cerebellum. Spine to cerebellum is an ascending pathway. So this is sending not motor information, but sending sensory information to the brain. Areas like the corticospinal, from the cortex down to the spine, are efferent pathways. And the efferent pathways that you would see, here we have the anterior columns, a bunch of clusters of axons that are traveling down. We also have pyramidal tracks to the side, also sending efferent information to the sides. Again, I'm not going to ever have you guys differentiate lateral versus anterior. And if you even go into the book, yeah, they go into more detail about each of these tracks. Don't worry about that. That's okay. The main message, efferent and afferent pathways. So the other circuits that we need to take a look at, here you see the spinocerebellar. There should be another one that is going to be looking at the ones that make its way over to the basal nuclei. So what exactly, again, a refresher of what the basal nuclei is responsible for. Its its responsibility is initiation and termination of movement. If I want to even start walking, that's going to involve the basal nuclei. Again, interneurons that are going to fire upon the local motor neurons to start the process. And then the stopping, again, is the basal nuclei. Now, on the other hand, the cerebellum is the one that's receiving information. And it's receiving information about what's actually happening. And the efferent pathway is sending efferent information about what the actually intended movement is. So now that we've laid that out, let's talk a little bit about the basal nuclei. The basal nuclei specifically looks at the initiation and the termination of movement. And any dysfunction of that, see bullet point number 2, suppress unwanted movements by inhibiting thalamus activity. So it works specifically on the thalamus of receiving sensory information, and then it can stop motion. So through this area, the basal nuclei has a specific area called the substantia nigra. The substantia nigra is involved with cell bodies that secrete dopamine. And dopamine is responsible for muscle tone. Okay? So it's involved in initiation and, again, termination of movement. Any regions, and Parkinson's is a typical area that affects this area, the substantia nigra, Parkinson's. That affects this area. And what you typically see with people with Parkinson's is a lot of unwanted movement. And due to the dysfunction of that area, they have the inability to stop movement. So their hands, as Parkinson's gets progressively worse, we start to see the basal nuclei, specifically the substantia nigra, that degrades over time and gets worse and worse. So typically with people with Parkinson's, we'll take dopamine in order to raise their dopamine levels. Now the cerebellum, like I said earlier, corrects any motion. So you have one motion taking place, that's sending information to the cerebellum through the spinocerebellar tracts, and then the cerebellum's going to say, well, if this is the motion, this is the actual motion, this is the intended motion, so we'll come in and correct what's taking place. So what the cerebellum does is it's going to receive and send information back to the motor neurons. So typically, if this were going back over here, and here we have the cerebellum, let's just draw the cerebellum back over here. Cerebellum is coming back and saying, hold on, wait a minute, this is what's actually taking place. Let's correct that and fire other neurons to correct the action. So typically, where would we ever see the cerebellum? Remember, when we saw the cerebellum, that was one of your quiz questions about the ataxia. Ataxia, if you remember that video of the guy could not walk in that coordinated pattern. And that's because he lacked any cerebellar action whatsoever to correct the motion. So what the cerebellum is supposed to do is send that information back to the motor neurons and say, no, this is the correct pattern. This is what you're actually doing. We want you to actually correct it. So if you were able to restore that cerebellar action as he's walking along, cerebellum will come in, correct it, and he would be able to walk normal. Okay? The other one was a horizontal nystagmus. This is the one where someone scans the room from left to right. If you remember, this is the story that I talked about, if you ever get pulled over and they want to look at your eye motions, what you should be able to do is move, and he has you track his fingers. You should really be able to smoothly track those fingers along. Alcohol comes in and affects the cerebellum. And if you affect the cerebellum, then you don't get that fine movement that you normally do. Your eyes will start to make this staggered movement because you can't monitor what your intended motion is, and you can't actually correct it. So your eye pattern does this. And therefore, knowing that your the cerebellar function is decreased, we can pretty much assume that's due to some form of dysfunction. And that's where they would go and actually do a breathalyzer test and find out, oh yeah, alcohol. Alcohol comes in. The reason we knew, there was a reasonable cause, we knew physiologically something was up with your cerebellum.
What if you're on a stimulant? That's still...
Stimulants can also affect too, depending on what stimulant that is specifically can cause excitatory.
Like cocaine?
Yeah, so excitation. They don't have to specifically look at the cerebellum. They can probably look at other features as well. If you're sitting there sweating and your eyes are fully dilated, you got another cause. So anyway, all it comes down to, and this is really not law, we're just looking at, they have reason to suspect, but when it comes down to alcohol, they know it's related to cerebellum. And then even look at people, look at cases where there's too much alcohol involved. Not only can you not follow the pattern with the eye pattern, you also exhibit a little bit of ataxia. So you see the guy that can't, or not guy, doesn't have to be guy or girl, that's walking along and they can't walk straight. Again, same thing, inhibition of the cerebellum. They cannot correct their actual intended motion. So you stop that feedback over to the upper motor neurons that are causing the contractions on the lower motor neuron side. So I know we had a question of amyotrophic lateral sclerosis, ALS. You guys remember the whole ALS craze that they had where they're like the ice bucket challenge? If anything, did kind of increase a little awareness. I would venture to say that even people really didn't know what ALS even was after the ice bucket challenge, but again, I'm digressing. So what exactly is amyotrophic lateral sclerosis? This is also known as Lou Gehrig's disease, because he was the famous sports person. I'm not into sports at all. Baseball, Lou Gehrig's disease, right? Baseball?
Yeah.
Baseball, okay, so that guy. So Lou Gehrig's was one of the celebrities that came down with this disease. And typically what is the main characteristic of ALS is motor neuron death. Okay, so it's actually death of the lower motor neurons. Again, going back to this right here would be the breakdown of the neuron itself that targets that muscle. And typically with ALS, some of the symptoms that you would see with ALS, especially early on, are muscle stiffening. You start to see the legs start to do more of a tripping action. So somebody with ALS can all of a sudden start tripping on one leg all the time. They can't really figure out what's happening with my left leg. It's like a bum leg. What is it doing? And then another characteristic feature is that they start to notice, especially as it starts to get worse, that foot isn't even stumbling anymore. It's now just starting to drag behind. And they're wondering, why is my foot not coordinated? So typically you would see it on one side. And as it starts to progress, not only are you going to start getting twitching, you're going to start excessive twitching taking place, and then later cases where you start to see atrophy. And atrophy is the breakdown of the muscle itself. Now that you decrease, you start seeing neuronal death. Anytime the neurons start to die off that target that muscle, and that muscle is no longer firing, you get atrophy. Okay? If you don't use it, you lose it. And that's basically what's happening in the case of ALS. Not only do they lose the ability to use it, is basically what it comes down to. Now, the etiology, the cause of the disease itself is unknown. I think they're still trying to figure out what causes it specifically. But what we know about it is that the disease starts around the age of 60. And once they diagnose you with ALS, unfortunately, the survivability is about 3 to 5 years. And it's a very fast process, because not only does it start with the leg, then it starts with the arms, and you watch people with ALS, and immediately one side of their body becomes very weak. You see major atrophy taking place, because then it starts affecting the other neurons. Then it starts working their way outside of the somatic, and starts to affect the autonomic. It starts affecting the diaphragm, and eventually they lose the inability to swallow, lose the ability to swallow on their own, to speak, and then they lose the ability to breathe. Like I said, the average survivability is 3 to 5 years. There are not a very small minority of people that last longer than 10 years. About 10% of those.
What's the name of the guy that?
Stephen Hawking. Stephen Hawking has survived it for about, was it 30 years now? 30, 40, 50, however old he is? It's crazy, he's had it for a long time. Multiple sclerosis is the other disease that you would have heard of. And multiple sclerosis is different in that it is an autoimmune disorder. This is where your own immune system starts affecting the not the neuron itself, but it actually affects the myelination. So typically with multiple sclerosis, it affects the neuroglia, specifically the Schwann cells in our peripheral nervous system and our oligodendrocytes in the central nervous system. So these start to break down and affect, they start to expose areas of the axon that have no Remember the myelin sheathing? Myelin sheathing in that area has no voltage-gated sodium channels. So once you start to expose that area, the axon the action potential comes in. It's supposed to jump to that node of Ranvier, but then it starts to fizzle into these areas that actually have no voltage-gated channels. So it starts to stop over time. So neurons stop firing down the axon. You start to, typically with people with multiple sclerosis, start to exhibit poor coordination, any sort of rhythmic activities they have difficulty performing. They do exhibit blurriness, loss of balance. Typically where you would see these symptoms take place, typically around the age of 20, 30, 40, 50, when you start to see people with multiple sclerosis. And it's actually 2 times more likely to happen in women than men. So with that, what you would typically also see with multiple sclerosis is scarring. We get the immune system, we get our own cells coming in and damaging the myelin sheathing. What you would also see is scarring that takes place that prevents healing of that tissue. Because what it comes down to is the neuron, the axon is fine. It's just the oligodendrocytes on the outside that need to come in and repair. And that scarring prohibits that. So in multiple sclerosis, they've looked into specific enzymes that can be used or drugs that can be used to come in and unscar those areas, to expose and promote oligodendrocytes to come in and wrap those areas. Again, really experimental. I think for the most part, they only work in mice so far. They haven't even gotten to the point of testing it out on humans yet. So the more we start to understand more about it, the more we can start finding cures for it. And again, there's no real cure for it because it's our own immune system that's coming in and attacking it. So we can only extend the life with those that have multiple sclerosis, but not necessarily cure them of multiple sclerosis. So that takes us into our reflexes. What we've talked about so far are the efferent pathways. We've talked about upper motor neurons, local circuit neurons. We talked about any neurons that innervate the local motor neurons to send information out to the muscle itself. But what we need to look at are the patterns that are exhibited by those inner neurons. And so we're going to start to take a look at some reflexes. These reflexes are things that are innate, meaning we've had them since birth. Some things we lose as we age, and some things we still maintain. So these videos really show you what the reflexes are all about. But before I start, reflexes typically happen. To give you a good example would be like a tickle reflex. It would come in, there's sensory information, I'm sitting there tickling the bottom of your foot. That's sensory information that's traveling over to your brain. And then efferent, it doesn't have to travel directly to the brain. It can travel to the spinal cord and travel right back out. So that is our reflex pathway. It involves sensory information going in, interneurons involved, and motor information back out. Things that we don't exhibit voluntary control over. The first video are all baby ones, and I think this is like 10 minutes long. I really want to get to some of the harder ones. I'm going to play some of them and skip forward. Again, really long. And there's actually 9 reflexes that take place. First one, blinking. So you see all these cute Facebook photos of people that are like, oh, look, my baby's winking at me. No, it's a reflex. Okay? It's not blinking at you, it's not communicating to you, it's just a reflex. The Babinski reflex. I'm going to pause it on this one. The Babinski reflex is something that you guys are going to do in... Oh, wait, sorry. The Babinski reflex is something that you guys are going to be able to do in a lab. In babies, what they do initially with babies, new infants, they come in and they want to test the reflex. What happens is they actually take any object and they brush along the lateral sides of the foot and they will stroke towards the medial side. What that's supposed to do in babies, in infants, is cause the Babinski reflex where the toes flare out. Okay? That is a reflex that occurs. This is sensory information going back to the spinal cord and then motor information going back out, causing the feet to flare out. Now babies do not have myelinated axons upon birth. And so what's happening is the upper motor neurons to the lower motor neurons are not fully developed in babies. So initially, the reflex that's taking place is purely lower motor neuron. That's what's happening with the foot. That's a Babinski reflex. Now, when you guys are doing the reflex on each other, what you're doing is to test that same pattern. And what you'll notice when you do that on somebody else is you get the opposite reflex. This is what we see, the reflex where the toes curl inwards. That curling inward is the upper motor neurons intervening on the lower motor neurons. And that happens after about, and I don't know how many months, maybe 4, 5, 6 months, when the myelinated upper motor neurons can now travel to the lower motor neurons that innervate the toes. So why they test this on people that have spinal cord paralysis? If they can still exhibit some motor control, right? One of the things they want to test is to feel and check for the Babinski reflex. On a trauma victim, if they stroke the the lateral side of the toe and it does the Babinski reflex on an adult, we know lower motor neuron, meaning from the spinal nerve to the toes, works. But we know what's been severed is the upper motor neuron down to the lower motor neuron. Okay. So something that they do on trauma victims. And in babies, they do exactly this reflex. Stroke right back out, toes flare outward. Again, lower motor neuron. Take a look. Oh yeah, this is the one where when you almost drop a baby, and they come in, they try and do a grasping thing. You see this in primates a lot. It's basically a reflex of when a baby's about to drop, they come in and they try and clench onto something. It's fun. If you ever have babies, try this on. Again, look, this appears in 3 to 4 months. Oh, this one's a fun one. This one's called rooting. This is an old video now that I look at it. Okay, so rooting. No, this isn't the fun one. Rooting is when you tickle the side of the mouth. They start to gravitate towards that motion. Again, purely reflex. Baby doesn't even know what they're doing. They just know what they're doing it. In fact, I don't even know if they know they're doing it. It's just instinctual. Again, a pattern that's taking place purely in the local circuit neurons, not motor neurons. Again, something that they do in order to initiate the sucking reflex, which is something again that takes place. Stepping. Oh, this one's my favorite one. These are the videos of baby videos that you see, and they'll take a baby, and immediately when the baby touches the ground, they're like, "Oh, look at my baby can walk. Such a good baby, so advanced." No, it's just a reflex. Baby doesn't even know what it's doing. Its legs are just doing this. That's all it is. This one, sucking reflex. Fairly obvious what that one is, so we'll skip that one. Got it. Great. Just keep doing it. Oh, this one's my favorite one. This is the video that you ever watch. You'll ever see photos of a newborn baby grabbing onto the finger. And you'll see that one where people are like, "Oh yeah, my baby knows it's me." No, it's just a reflex. So feel free to say it on my behalf. Marlon said it's just a reflex. Be that buzz killer.
Thank you.
I'm in my 30s. I'm gonna see all these baby photos coming out. I'm like, come on. Not that I like babies. No, I hate babies. They creep me out. But yes, there it is, grabbing onto something. Purely reflex. Won't let go. Cool. Other ones, swimming. There's a bunch of other ones. Swimming. This one's always an interesting one. Best time to get a baby to learn how to swim, utilize that reflex. Throw a baby in the water and watch them swim. So swimming schools will typically talk about this, and they'll say, oh, you utilize that swimming reflex because they start to lose it at 6 to 7 months.
That
is so
cute. This earlier.
Yeah?
Yeah, she said her baby went swimming because she had a pool in her backyard. Then now she's teaching them how to swim.
Yeah, and actually they will build on top of that because now they start to learn the motions. The more they do it, they realize, yeah, they can swim.
Yeah.
They've only been swimming for like 9 months.
Does it hold its breath? Or?
What's that? That's actually another reflex too.
Oh, okay.
You blow into their face or anything like that. And if you ever watch babies learn how to swim, the first thing they do is blow right into their face because that'll cause them to hold their breath. Then they dunk them in, and then they'll initiate the swimming reflex. And they'll swim, and then they come right back out. You guys ever use that with dogs too? You ever put dogs in the water?
Yeah.
Dogs don't know what they're supposed to be doing, but they're all of a sudden just doing this.
Yeah.
They know what they're doing. Reflex. And tonic neck, these ones are not as fun. Tonic neck. That one, another weird thing is if you've turned their necks like this, they'll, and then you turn their neck, the arms will just flip over the other side. I don't know. Again, reflex. All right, so the other ones for adults. Now that we get to see 10 minutes worth of baby, let's look at like the other 2 minutes of adult. Things that we retain from infancy. Oh. I knew I wanted to plug this in for a reason. And these are the things that you guys are going to be performing on yourselves as well.
Examine in a patient for a screening neurologic exam. We will.
So one thing I love about videos is how long it takes to just get to the point. And here we're going to talk about the knee-jerk reflex.
Thus eliciting a
not knee-jerk.
symmetrical reflex in this individual.
But the reflex, what they're
The
doing here
triceps reflex
is
is
they're
more
striking
difficult.
against the brachioradialis.
Some positions that can be helpful in
strike will
eliciting the
result in a
reflex
muscle
are to have
twitch.
the patient dangle their forearm and then to palpate the triceps tendon and strike over that tendon. Another position that helps to get the arm into relaxation.
Okay. This one is the one that we're going to be able to look at.
The knee jerk is
Knee-jerk reflex.
elicited by striking over the patellar tendon just below the patella.
And punch. So we'll talk about why that reflex takes place.
And the Achilles reflex elicited by striking over the Achilles tendon. It's important to make sure that the foot is partially dorsiflexed so that the tendon is partially stretched before trying to elicit the reflex. If you have difficulty eliciting the reflex in that.
Great. So those are the reflexes, again, that are carried from birth. So looking at these reflexes, you're going to be able to see we can classify them. Not really the entire heart of what we want to talk about, but really we can classify reflexes by their development, whether they're something that they're innate or something learned. Where does it take place, whether it's specifically in the spinal nerves or the cranial nerves itself. And then the motor response. Tons of types of reflexes. All you were looking at were the skeletal muscular reflexes. But there are reflexes that we'll be talking about that take place in the visceral organs themselves. Something that happens as a response, sensory information is sent in, and a motor response is elicited afterwards. And then the next thing we're going to be able to look at is the complexity of the neural circuit. Do we involve one synapse, or do we involve two? So the types of reflexes that you can see here, the ones that we're going to be really looking at, are the somatic reflexes. Control skeletal muscle contractions. This is the heart of what we're going to be talking about in today's reflexes. I've got 3 types of reflexes that we'll cover. The other visceral reflexes are things that we'll cover as we go through. We'll talk about certain reflexes that take place. A good example of that is the swallowing reflex. Whenever you take, you're consciously chewing, and then when you swallow, that is a reflex. What it's going to do is going to lift the epiglottis, and it's going to allow food to travel down the stomach. Completely reflex. All right, so looking at the spinal reflexes, we're going to be looking at the stretch reflex. The stretch reflex, here you can see, is going to involve two types of a monosynaptic and a polysynaptic component, and I'll go into more detail about that specifically. And then the next thing we'll look at is the muscle sensory receptors. Where the receptors are located, we'll talk specifically about a muscle spindle receptor and a Golgi tendon organ receptor. If you take a look at the middle slide right here, the middle image, you're going to see that we have muscle spindles that run parallel to the muscle fibers. And what they're going to be doing is going to be sensing the degree of muscle tension that's taking place, how much contraction is taking place on that muscle. Now the other one that we'll talk about is the Golgi tendon organ. And the purpose of the Golgi tendon organ, and it's located just, if you were to look at the name, the Golgi tendon organ is in series with the muscle, and it's located on the tendon itself. Again, this is going to tell us the amount of tension, not on the muscle fibers, but specifically on the tendon itself. And this is going to send information back over to the spinal cord. So looking at the first one, we're going to see the first one called the muscle spindle reflex arc. And the best example of this is the knee-jerk reflex. So typically in the assessment of this reflex, what they would do, and you guys again, we're going to be doing this on each other, you'd be sitting at the edge of a table, and you're going to let your foot dangle, and they're going to use a patellar tendon hammer, a patellar hammer that's going to strike the patellar tendon. And what that's going to do, it's going to cause a contraction. So the sensory information that's being sent in, when we strike with the patellar tendon, that causes a stretch of the muscle. So strike that tendon, that's going to pull and stretch the muscle itself. So that stretching that takes place is going to send that information back in, and what it's going to do, it's going to target the lower motor neuron that targets the quadriceps. So what starts off with an initial stretch results in an excitation of that lower motor neuron. And what does that excitation do? Causes contraction of the quadriceps, hence the kicking action. Okay? So that should make sense. Stretch results in a contraction to limit the amount of stretching that's taking place in the first place. So this, in effect, is a form of negative feedback, where we start off with a stretch, results in a contraction. Now, think about what that means. If I'm going to contract the quadriceps, would it do me any good if the hamstrings contract at the same time? Because that hamstrings is a antagonistic muscle, meaning it's going to counter the contraction taking place. So if you activate the quadriceps, what we need to do is relax the hamstring to prevent that hamstring from firing. So the same pathway, the same sensory pathway that comes in that excites the quadriceps also targets an inhibitory interneuron that hyperpolarizes the motor neuron, or makes it less likely to fire, on that hamstring. So this is an illustration of a monosynaptic pathway, where the monosynaptic pathway is the sensory neuron traveling directly to the lower motor neuron. There is no interneuron involved for the excitation of the same muscle. Okay, does that make sense? One monosynapse, one synapse, no interneuron traveling to. So all of this reflex is taking place on the ipsilateral side. And that's a term, ipsilateral, that I'd like you to be familiar with. Ipsilateral means everything that's taking place on the same side. If I hit your left patellar tendon, it doesn't affect your right leg at all. So this pathway travels all through the left side of the spinal cord. Both excitation of the quadriceps and inhibition of the what we call the antagonist muscle, inactivation of the antagonist muscle. So this portion right here is not monosynaptic. This is polysynaptic in that it involves an inhibitory interneuron. Because just like I said earlier, the only way I can make this motor neuron, which is responsible for this hamstring, the only way I can get this to be less likely to fire is through an inhibitory interneuron. In each of our diagrams, you'll be able to see sensory information in blue and efferent in red.
So antagonist, you need an interneuron, right?
Yes, if you, for the antagonist, if you're going to inhibit the antagonist muscle, remember to inhibit a muscle, you can't, there is no inhibitory neurotransmitters on the sarcolemma, or the neuromuscular junction. You have to inhibit it on the cell body, which is located on the dorsal gray horn. I'm sorry, the ventral gray horn. Okay, what questions do we have about this reflex arc? It's one of our simplest ones. What starts off with a stretch results in a contraction of the agonist muscle, results in a relaxation of the antagonist muscle. You don't have any questions now because I'm going to move over because this gets a little harder. Good? Okay. Golgi tendon reflex arc is considered polysynaptic. The whole purpose of the Golgi tendon reflex arc is to limit the muscle contraction for anything excessive. Now take that 100 pounds that I'm curling and it's way too much. And I'm straining and I'm straining and I'm straining. I want to prevent muscle damage. So what's going to take place is inactivation of a muscle that's overly contracted. So what a Golgi tendon does, and take a look at where the receptors are located. Again, on the tendon itself, which is going to receive information about how much tension is on the muscle itself. So what that's going to do, it's going to send information back. So we're contracting that muscle. The Golgi tendon organ is going to send that information back, again through the dorsal root ganglion, through the dorsal root into the dorsal gray horn, and it's going to send an inhibitory interneuron to inhibit that same muscle which had the Golgi tendon organ. So what started off as a contraction results in an inhibition. So why was it not direct? Why did we see it's polysynaptic? Because what I'm doing is inhibiting that contraction. The only way I can do it is to involve an inhibitory interneuron. Now, the other way, when we look at the antagonist muscle, where we see relaxation of this muscle, we want to also see contraction of the antagonist muscle. Because what that's going to do is if you see contraction out here, that's going to pull the muscle back out. And now you want to use the hamstring to flex the knee again. So in order for that to take place, that same sensory neuron comes in and targets an interneuron that comes in and excites another lower motor neuron. So we use another excitatory interneuron to target another local motor neuron that directly innervates the hamstring. So what the primary result, again, just a recap, what started off with excessive contraction results in relaxation of the agonist muscle and results in excitation or contraction of the antagonist muscle. How are you guys feeling about this one? Yes?
Curious, like, how does your body know that? Like, because when you weightlift, you could rip your muscles, couldn't you? If you just go too fast, or?
You can through the upper motor neurons because your intent is to continue that contraction. But eventually, this becomes a fail-safe. This fail-safe is completely innate. It tells the feedback that comes in that says this is just too much, and the muscle fails at that point in time. And that reflex takes place and relaxes it. Give another good example. Is a good stretch. When you notice a stretch, what you're doing is contracting. You're actually stretching the muscle, which is resulting in a lot of tension in that Golgi tendon organ. What do you notice about it when you're in a stretch position and you hold that position for a little bit longer and a little bit longer? Don't you notice you kind of sink into it a little bit? No? No? None of you guys do any stretches? Well, do some. So what you notice about certain stretching is when you put a good amount of load on that muscle itself, it relaxes after about 5, 10, 15 seconds. And all of a sudden you notice, oh, I got into that stretch a little bit more. The reason being, you have that Golgi tendon organ that comes in and reduces the amount of contraction that's taking place in the first place. It inactivates that same muscle that's being stretched. So what starts off as a certain amount of tension, you get inhibition on the spinal cord level.
So
Yes.
what would be the difference between this one and muscle reflex?
The first one, muscle spindle, is where we had the initial stretch of the muscle.
Uh-huh.
That resulted in contraction.
Yeah.
This one is contraction that resulted in relaxation.
So then the antagonist changes here?
Yeah, in the antagonist, we see the exact opposite. Where here we saw relaxation of the antagonist, we see contraction of the antagonist. So here, the main idea behind this is that anytime you get a stretch of the muscle, you want to like any unwanted stretch of the muscle, you want it to contract back in. You want to prevent that muscle contraction for the first place. Well, this one, too much load, stop it. Any other questions before we move to the last and final reflex? Because this one gets nasty, right? So this, take a look at the patterns on this. There's a lot going on here. This is my favorite one. This is called the Flexor withdrawal reflex. Some of the key things that you have to pick up from the Flexor withdrawal reflex is that there is a response that's happening due to some form of stimulus. This stimulus is how many here have ever stubbed their toe? Really, like 3 people, 4 people? All of you have at some point have stubbed your toe on something. And when you stub your toe, whether you stub your toe or you step on a nail or something like that, this involves a reaction that takes place on one side, which is typically pull your foot away from the stimulus, but another response called the contralateral response, which happens on the other side. And not only do you see responses happening on the other side, you're also going to see local circuit neurons. So let me first start off with what's happening. I'm walking along, and I'm going to stub my right toe. What do you think when I stub my toe? What motor response do you think I'm going to do?
You pull it back.
I'm going to pull back my right toe, or pull back my right foot. But if I pull back my right foot and I don't do anything else, I'll fall over, right? So what's happening when I pull back? This is the flexor withdrawal. I'm withdrawing that leg. What's happening to the other side is I'm stabilizing everything that's taking place on the left side, on the contralateral side. So right side will withdraw, left side will contract. So in that case, I need to involve both sides of the spinal column. So to play back, we'll first talk about the right leg. The sensory input is received that you had pain on your right leg, and your instinct at that point, so here the sensory information is being sent to the spinal cord segment, the middle one here, spinal cord segment that innervates the interneurons that are involved in withdrawal of the leg. In this case, this would be the hamstrings to withdraw the foot away from the stimulus. So sensory information goes in, recruits our interneurons to cause activation of the hamstrings. Not only does it activate one motor neuron of the hamstring, but it also is going to use our local circuit neurons to activate the spinal cord segment below and the spinal cord segment above that innervate the same muscle. So now you're going to see recruitment. And this, in a way, is a form of positive feedback where I'm now recruiting the other local motor neuron that targets the other muscle spindles below and targets the other muscle spindles above. So this is an exaggerated response. What starts off with an initial, if I only had one interneuron, I might get a slight withdrawal. Now if I get other motor neurons involved, I'm going to get an exaggerated withdrawal reflex. Okay. Now when I get that reflex that's taking place, notice what that sensory neuron is also doing. It's recruiting another interneuron that travels to the opposite side through the gray commissure. Remember, that area that allows crossover from one side to the other. So as it crosses over, here you can see this is recruiting, again, it's an excitation of the other contralateral muscles, specifically the quadriceps. The quadriceps are now flexing in order to stabilize my left side. So not only is it stabilizing the quadriceps, it's also going to, again, use local interneurons to not only innervate the quadriceps and with that motor neuron, but all the other local motor neurons that also target the muscle spindles for the quadriceps. So here again, you get the exaggerated response. Much like what you saw on the hamstring, you see the same thing happening in the quadriceps of the opposite leg. So if you don't believe me, try it at some point. Try stubbing your toe. You'll get a withdrawal on one side, and you'll get stabilization on the other side. Not only do you see the quadriceps, you also have the hip that's involved. You have the hamstrings that are involved, really to balance yourself on one foot. And all of that was reflex. Did you ever think, oh, I just stubbed my toe. What should I do? You don't have your upper motor neurons come in and say, oh, Marlon, why don't you flex your hamstring so you can pull away? Great, oh, but don't forget, use your other interneurons to come in and stabilize your other leg. No, it's complete. You did all of this before you even recognized you stubbed your toe. At some point you knew, oh yeah, I stubbed my toe. Sensory information is getting sent in not only to the interneurons that are involved in the motor pathways, but you're also getting sensory information traveling up those tracks back over to the cerebral cortex. And the areas that are lighting up, the sensory area, which is the sensory motor cortex, specifically the sensory area, which is the most anterior region of the parietal cortex. The sensory, if you remember the sensory motor homunculus, that area is lighting up, saying, yes, I receive pain in that area. So here you can. These are the reflexes that take place, but you could override them. Your upper motor neurons can come in and say, nope, don't do that, and do exactly what you don't want it to do. But for the most part, these neurons, why is it a reflex? Because all this takes place faster without actually involving the brain itself. That's why they're reflexes, because they happen specifically in the spine.
So does it send it to the brain? It does, or does it?
It does, yeah. Eventually, this interneuron travels up. This sensory neuron travels up, and it's going to send information back up to the brain. So if you ever stub your toe, you notice that you're able to stabilize yourself before you even notice you even stubbed your toe. All that happened because this is literally a local circuit. It's fast. The time that it takes to go this takes much less time than it takes to go than up to the brain.
The brain makes curse, right?
Yeah, then all the motor stuff that happens with your mouth and fingers and whatever else you do, that's something that happens after. So this is the entire reflex that we just mentioned, and this happens in quite a bit. This is all that's taking place. You can also see the same thing happening whenever you touch something. If you ever tested the whole thing about touching a hot pot, you touch yourself and there's that pain withdrawal, same thing. That happens on one side. This might be more of a ipsilateral, meaning all happening on one side, maybe not so much happening on the other arm. You're not really doing anything else with the other arm, but same thing. This is the pain withdrawal reflex.